I think I will save my the discussion of the real project for whenever the seminar happens. But today I thought I would instead try to give you a little bit of an introduction to me, where I come from, and a little bit of an overview of some of the research we do, just to give you kind of a concept. Of course, Virginia has already given you a very good general introduction. So on a personal level, I started life in Argentina, so my sort of cultural reference points are Borges and that whole Latin American literary flowering that happened in the 1960s, 70s, 80s. And, of course, tango and pistola, who's very dear to my heart. And, of course, Malbec, which is another excellent product from Argentina. And it's nice to be in France, where I can also compare French Malbec and Argentinian Malbec. The Argentinian is much better. But all the other kinds of wine are much better in France. So that was where I started. In terms of my education, I did all of my university training in the United States. First of all, at Boston University, where I did a bachelor's degree in music and in psychology. And then I followed it up with my PhD in experimental psychology at Brown University. And something that was interesting, my experience in Boston, was that at the time anyway, to do a degree in music and psychology meant that there was not one course in common. So I had to do all of the coursework in music and all of the coursework in psychology, which was great because I got two things for the price of one. However, I noticed at some point that when I was in my music classes, all of my friends were actually a lot better than I was in whatever it was, music theory or history or performance. Whereas when I was in the psychology classes, I was doing pretty well, and I didn't feel like I was just mediocre. So that's why I decided to go into more of the science part. And this is Brown University in Rhode Island, in Providence, Rhode Island, close to Boston. But I didn't want to give up on music, and my training was actually in the organ. So really I tried to put my two passions together and try to understand a little bit more about music from a scientific point of view. So to really try to do that, to try to put those two things together, I decided I needed training more in neuroscience, and so that is why I went to Montreal to do my postdoctoral training there with Brenda Milner, who's a very well-known scientist. And I was lucky enough to be able to get a faculty position there in 1985, and I've been there ever since. I'm currently the head of the Laboratory for Auditory Cognitive Neuroscience, and I'm also the co-director of BRAMS, which is the International Laboratory for Brain, Music, and Sound Research. And that's co-directed with my friend Isabelle Peretz, who's actually Belgian but has been in Canada for a very long time. And I like to put up this image because that's our building there, the Neurological Institute. It's usually covered with snow, but this is a few days in the year when it's actually warm. But right on the side of the building is this statement from the founder of the Institute, Wilder Penfield. And what he said is interesting to me. He said, the problem of neurology is to understand man himself. La neurologie cherche à comprendre l'homme lui-même. And, oops, what happened? Here we are. So maybe in slightly more contemporary language, I would restate that to say what neuroscience is trying to do is to understand what it is to be human. And in that context, I think the research that we do on music fits into that broad idea, that I would argue that to be human is to have music and to have art and to have literature and to have science and to have a lot of interesting things. So I don't see a schism between science and art. I see them as coming together. Now, by way of introduction, I think Virginia has already given away a lot of the concepts here, but why do we study music? What's interesting about it? Well, there are many interesting aspects. One of them I find is that it's actually a very ancient aspect of our species. It is not a recent construction, like, for instance, alphabetic writing, which appeared somewhere in Mesopotamia, perhaps, or in the Middle East a couple of thousand years ago. Music is actually much more ancient than that, as far as we know. And part of the evidence for that comes from archaeological work. I'm certainly not an archaeologist, but I love to talk about it. For example, these flutes that you see here were found in a cave in China, and they've been dated to something like 5,000 years ago. What's really amazing about them is that they can still be played. And if the audio works, I just want to play you a little excerpt. It's absolutely beautiful sound, right? And this is made from the bone of a wading bird, like a stork or something like that. So that it's not that ancient; it's 5,000 years old. But remarkably, even more ancient flutes have been discovered recently in what is now the Danube Valley of southern Germany, or into Hungary. And these are from the upper Paleolithic period, so they're about 40,000 years old. And they're, again, very beautifully engineered. You can see the holes are a certain distance apart to create a certain scale. They're even beveled so that your fingers will close the hole nicely. To me, what this means is that already at this time, there was this remarkable technology to produce music, which means that music must be much more ancient than this, right? Because things like singing or clapping or dance, there are no fossils remaining from that, but clearly in order to have something like a scale system, you must have had a development of music that occurred over presumably many hundreds of thousands of years. So I would argue that music is sort of part of our original human mental machinery, and therefore must have a kind of biological basis, and what we are trying to understand is that biological basis. Everybody knows this statement of Darwin's about music. In The Descent of Man, he said, "As neither the enjoyment nor the capacity of producing musical notes are faculties of the least direct use to man, they must be ranked amongst the most mysterious with which he is endowed." So he sort of threw up his hands and said, I have no idea why we have music. It doesn't really make sense. But there's a less well-known statement that he made in his autobiography many years later, near the end of his life in 1881, and he said this: "If I had to live my life again, I would have made a rule to read some poetry and listen to some music at least once every week, for perhaps the parts of my brain now atrophied would have been kept active through use." Which is really remarkable, right? It's a very insightful statement. And then he says the following: "The loss of these tastes," because he was actually very depressed later in his life. You know, his favorite daughter had died quite young. He was not a happy man. And he says that he no longer enjoyed music or literature. And he says the loss of these tastes is the loss of happiness, may be injurious to the intellect and to the moral character by enfeebling the emotional part of our nature. And I think here he actually answers the question that he was posing before. This is the reason why we have music, is because it allows us to communicate, and in particular to communicate emotions. So the approach that we are taking, I think, as was already mentioned by Virginia, is to really understand human cognition by looking at studying the anatomy, the connectivity, the functional properties, the plasticity of the brain, and in other words, to understand this biology behind it, which we think is very relevant. What are some of the methods that we use? I'm going to just take the last 10 minutes here to tell you a little bit about some of the approaches that we take. So as I mentioned, I was trained in experimental psychology. What that means is that we always are trying to explain and understand behavior. We're understanding behavior from the point of view of the brain that produces the behavior, but the goal is actually to explain the behavior, which means that we need tools to measure behavior. So this is my lab. On the right, you see one of our soundproof booths where we have people sitting there and listening to different kinds of sounds, making judgments about them. We measure reaction time, we measure accuracy, things like that. On the left is another room where we also measure psychophysiology. So we measure things like skin conductance, heart rate, respiration, things like that. I managed to obtain some funding to buy myself an organ, which is very cool. So I get to play in my lab every day. Actually, almost all my students, as Virginia students too, are mostly musicians. So on the weekends, I have jam sessions. They all play different instruments. I tried to jam with the organ once. It just didn't really work at all. But the organ, apart from being a magnificent instrument, is of interest also from the perspective that it requires a very particular kind of coordination. It is the only instrument that has full control over the feet. You see on the bottom is the pedal board, and the pedal reproduces 2.5 octaves of the, in other words, the same structure as you see on the manuals on the keyboards, which means that you can do things like play a certain tune with your hands, but you can also play the same tune with your feet. And that raises interesting questions about what is the mental representation of the motor actions that allows you to transfer from one to the other. In fact, if you're a good organist, which I'm not, you can actually sight read, let's say, a polyphonic piece and you can exchange the tenor voice with the bass voice, play one with left hand or play one with the feet. You're supposed to be able to do that. Second piece of technology that we have are things like electroencephalography, which is something you may have encountered. This is where you measure the electrical fluctuations that occur from the brain that is inside your skull. The electrical changes are very slight because your skull tends to attenuate them, but you can still measure them with the right wires. And even better is the magnetoencephalography, which looks like a gigantic hairdryer. That's because it's supercooled with liquid helium, and the supercooling allows you to pick up the very, very weak femtotesla fluctuations in magnetic activity that are actually produced by the neurons in your head. This is especially important for us because it means that we can pick up brain activity with very precisely in terms of time. Not so much in terms of where in the brain, but in terms of the temporal precision. So we can generate lovely images like you see here. The reconstruction of the image on the right tells you the approximate location of the waves. And on the bottom, we see one of the kinds of brain waves that we can measure. What's interesting here, this thing is called the frequency following response, FFR. It actually is a brain potential. It's a fluctuation in brain activity that, as its name suggests, follows the frequency of the sound. So if I play you a series of tones, do, 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 you would have something in your brain that actually follows the frequency that is introduced into the ear. So that's a very precise kind of measurement that we can do, and it's of interest to us because it is modified, for example, by musical training. So if you're a musician, that thing looks a lot more like the real sound. If you're not a trained musician, you still get that frequency, but it's more unstable. That, in turn, is of interest because, for example, it is related to how well you can discriminate different pitches. So here we see a correlation between the amplitude of that signal in the brain, shown on the y-axis, and your ability to discriminate two tones that are very close together in pitch. So this has direct consequences, and I like to show this because it's the link between the brain activity and the behavior. Remember I said we're always trying to explain behavior, so in this case the behavior would be what is your capacity to discriminate fine pitch. That's what we can measure behaviorally. We can explain some proportion of that variance, about a quarter of it, by looking at how your brain is encoding that sound. Oops, yes. So another piece of technology that we have is MRI. As Virginia mentioned, we can use it to look at brain anatomy. This is how it's used clinically. If you have a disturbance in your brain, a tumor or a stroke or a vascular problem, you would be able to see it. We use it slightly differently. One of the things we can do with it is to measure cortical thickness. So these images here, usually you see these colored images that represent brain activity. In this case, colored images represent the thickness of the cortex. So we can take the MRI scan, then we apply mathematical models that model the surface, so all of the dips and grooves and so forth that the brain has. And from that, we can calculate how thick any given spot is in the cortex. And what I'm showing you here in the colors are the areas of the brains of musicians that have thicker cortex than those of average people of a similar age and similar education and so forth that don't have musical training. And it's of interest because you can actually see that there are changes over here in these auditory areas. Oh, you can't see my cursor. Anyway, there are changes in a number of different parts of the brain. So that's of interest in terms of plasticity, so how the brain adapts to the task that you give it. Here's an example of functional imaging. So now the color scale actually refers to the amount of brain activity rather than to the brain anatomy. And somebody was asking about singing earlier. So here, in fact, we have trained singers who are actually vocalizing inside the scanner, and we can see the specialized network that is associated with singing. Here you can see a little bit how precise we can be in terms of the location of the activity. And the big yellow blotches there are actually representing the part of the motor cortex that is responsible for controlling the larynx. And of course, larynx is obviously involved in singing, and that area is typically more highly developed in people with a lot of singing experience. And still sticking with the functional imaging, possibly the work that we're most well known for is a series of studies that we've done looking at the functional response, the brain activity pattern associated with very pleasant musical experiences. So we've done a series of studies, and we always see these deep brain structures. So not the cortex, not the outer part, but deep in the subcortex, we see some regions that are very consistently activated, in particular one region called the striatum. And what's remarkable about that is that other people have demonstrated that the striatum, is shown in the top row here, is always active for things that are biologically important. So for example, if you do experiments with food rewards, like shown in the middle, so you scan people when they're either viewing, they're very hungry and they're viewing pictures of delicious food, or they're actually being delivered little bits of food. You see that structure. Also in gambling tasks, so you play like a gambling game and you get money and it's very rewarding, it's very exciting, you see the same structure. And in the final column, erotic rewards, these are experiments where people are watching erotic movies and they're very arousing, and it's always the same structure. And we see that same structure with music, which, like I said, has given us a lot of publicity and interest. Another technique that we have is brain stimulation. This is especially interesting because not only can we measure the brain activity, we can actually change it. We can do this in various ways by using a coil, for example. We pass a very strong current through this coil. The current results in a change in magnetic flux, goes through the skull, activates the brain. And one thing that we've done with it is we have actually been able to improve people's memory for sounds. So if you target the exact right area with the exact right frequency at the exact right time, which is not easy to do, we can actually get a boost in memory performance for the ability to retain these little notes. And that, of course, is of interest because it might have some eventual clinical application. So what am I doing at IMéRA? First of all, I am going to pursue some research projects with Pascal Belin, who's at the Institut de Neurosciences de la Timone, He is one of my former students, actually. Was in Montreal back about 20 years ago now, and he has a very, very distinguished career of his own. And another colleague, Benjamin Marion, who is at the Institut de Neurosciences des Systèmes, and I have no idea what the difference is between the two of them, because they're just like in different buildings, both at La Timon. But we're going to try to develop some research on acoustical markers of emotion and music, and I'll maybe talk more about that when I do my seminar. And possibly looking at some recordings that they have available from patients. And finally, I'm writing a book which is tentatively entitled From Perception to Pleasure, not Pleasures, The Neuroscience of Music and Why We Love It. And I'm currently dealing with some publishers. If any of you, I've never written a book before, I have no clue how it's really supposed to happen. If some of you have written books and can tell me how I should negotiate with MIT Press versus Penguin, please tell me what I should do. Merci beaucoup.
But does it have potential or actual results in improving memory period?
Yes, I think so, because we're just using, in that particular experiment, we're using musical tones because we can manipulate them very easily, and we know exactly how to look at that. But we don't think when we stimulate, we don't think we're stimulating circuits that are unique to those particular sounds. We think we're stimulating circuits that would be useful for other kinds of stimuli as well. So the hope is, although we don't have the evidence yet, the hope is that if we do this, right now one of my students is trying to do this over a period of a few weeks. In other words, do the stimulation, spreading it out over many weeks. In the first experiment, it's only 1 day. And then we want to see if it lasts. And if it does last, then we can test, does it extend to other things than music. I would certainly expect it would extend to other sounds, like speech sounds, for example. If it would extend to visual stimuli, I don't know, but that's a whole research theme that we hope to pursue.